let's discuss about the another and most important secondary messenger GQ and phospholipase C beta, which will give rise to IP3 and DAG, which is another very much important secondary messenger. But in NEP curriculum, NEP syllabus of University of Calcutta, the secondary messenger IP3-DAG is not included. But if you want to study the signal transduction or cell signaling in some detail for your future purposes, it will surely help you. Okay, so now let's concentrate about another second messenger. G protein is now activated with binding of signal molecule with GPCR, a G protein coupled receptor. After this step will be formed, and this GQ and phospholipase C beta, I have already discussed that this is another major effector protein of G protein. Phospholipase C beta, this one is also plasma membrane bound like CAMP that I am already discussed previously. Okay, this is also plasma membrane bound phospholipase C beta. Here the G protein specifically named as GQ. Here the G protein is specifically known for this phospholipase C beta is GQ. Normally, mammals express 4 classes of phospholipase C beta: PLC, delta, PLC gamma, PLC delta, PLC epsilon. All of these 4 types of PLCs require calcium ion for their activation. It requires calcium ion for their activation. The activated phospholipase, it's now activated phospholipase C. I am not going to specifically where is this beta, gamma, delta or epsilon. Normally phospholipase C is now activated. It cleaves phosphatidyl inositol 4,5 bisphosphate. Phosphatidyl inositol 4,5 bisphosphate, PIP2. After this cleave, phospholipase C beta cleaves this phosphatidyl inositol 4,5 bisphosphate, and what will going to generate after this cleavage? It will give rise to IP3 and DAG. Full form inositol 4,5 bisphosphate and DAG full form diacyl glycerol. These two act as second messenger. Watch it very carefully, and this step is very much important. Phospholipase C beta, activated phospholipase C cleaves phosphatidyl inositol 4,5 bisphosphate into IP3 and DAG. Full form of DAG is diacyl glycerol, and IP3 full form inositol 4,5 bisphosphate, and both of these two act as second messengers. This PIP2, this one is membrane phospholipid. This one is membrane phospholipid, plasma membrane phospholipid, and it is a minor component of plasma membrane, localized to the inner leaflet of phospholipid bilayer. Its location is inner leaflet of phospholipid bilayer. One secondary messenger DAG is remain associated with the plasma membrane. This remain associated with the PM, plasma membrane. And the another secondary messenger IP3, which is also produced from after cleavage of PIP2. This is a small polar molecule released into the cytosol. This one is released into the cytosol. This one is attached with plasma membrane and this one is released inside the cytosol. It gives signal, and after giving this signal, what happens? Release of calcium ion from endoplasmic reticulum, and for this reason cytosolic calcium level arises. Cytosolic calcium level arises, and this elevated cytosolic calcium level affect the activation of variety of target protein like protein kinases and protein phosphatase. So these protein kinases and protein phosphatases are final target protein of this IP3. For this purpose, for this release of calcium from endoplasmic reticulum, IP3 bind with receptor. This cause ligand gated calcium ion channel is being opened. Let's concentrate and sum up. This secondary messenger IP3 is released into cytosol. It gives signal, and for giving this signal, calcium ion is released from ear, and for ear means endoplasmic reticulum, and for doing this another step is performed by this IP3. It binds with a receptor, and for this binding a ligand gated calcium ion channel is being opened, and finally calcium ion is released from endoplasmic reticulum. Then obviously cytosolic calcium level is being increased, which cause the activation of its target protein kinases and protein phosphatases. So this is how IP3 secondary messenger works. Okay, now come to another secondary messenger working principle of secondary messenger, diacyl glycerol. Phosphatidylserine with lots of calcium ion. Okay, and this one is also activate protein kinase C. Once PKC get activated, what have PKC done? PKC is being activated and phosphorylate serine threonine residue of target protein. Okay, attached with phosphatidylserine in presence of calcium activate protein kinase C, and protein kinase C after being activated phosphorylation of serine threonine residues of target protein occurs. So this is working principle of IP3 and DAG. Now let's concentrate about the diagram of forming IP3-DAG. All of you know that this is plasma membrane and this is protein lipid protein layer of plasma membrane. Let's write here plasma membrane, and this is protein lipid protein bilayer. This one is GPCR. Ligand binding is already done, and here it is being activated. Here G protein alpha is now bind with GTP, means this is this, and here beta gamma separate. Draw it separately because it is activated now. Specifically G protein is GQ protein, and this one is activated now. PLC beta. This activated G protein is going to activated PLC beta. It gives rise to, it's going to cleave PIP2, and PIP2 give rise to IP3-DAG. This DAG here activate PKC Okay, it's going inside and open ligand gated calcium ion channel. This is ligand gated calcium ion channel. This channel is open now and calcium is released. Okay, calcium is released and this calcium is going to activate this process, which I have already give a description to you. And here this one is ear, endoplasmic reticulum, from which this calcium, ligand gated calcium ion channel is being opened and calcium is being released. Here lots of calcium are there, and this one is ligand gated calcium ion channel. So this is diagram of IP3 and DAG. Okay, so another second messenger, IP3-DAG, and its working principle with this pointed diagram is being completed.